This podcast is brought to you by the School of Advanced Study, University of London. All of our podcasts are available from our website, www.sas.ac.uk.
Barry, Dr. Chandaria, thank you very much. I was enjoying listening to your lecture much more than I think you'll enjoy mine, but there we are. I'm well aware that the title of this series is more than a little inflated and ambitious, particularly in this environment surrounded by experts in the humanities, people who think very deeply about the characteristics and the potential of the human mind. However, if, like me, you are essentially a materialist, if you believe that what's inside our heads gives us the capacity to do the things we do, then we have to account for a lot of the things that we understand at both the sophisticated philosophical level and also at a lay level in terms of folk understanding. We have in the end to give accounts of those things in terms of the activities of this thing. This is Wilder Penfield's photograph of the human brain, of a living human brain in a patient with the skull, a large section of the skull removed to expose the brain. The patient was conscious. The operation was done under local anesthetic. Penfield was intent on removing part of the brain, which in this patient caused massive seizures very frequently. As a surgeon, what he was trying to achieve was to remove part of the brain that was causing seizures in this person, massive epileptic seizures. But he didn't want to remove any part of the brain involved in some absolutely crucial function, particularly language. So he performed a preliminary procedure, and you can see the elements of it here, exploring the brain, exploring the area of brain around the region he wished to remove, to see whether electrical stimulation of it might cause symptoms, mental experiences in the patient, or temporarily inhibit actions of the patient, like moving their hands or speaking. Now, I realize that I'm probably talking largely to an audience of non-biologists. For your sake, I should say that the brain doesn't come with the little numbers on it. It'd be very useful if it did. These are marks that Penfield's put on to indicate where he had stimulated, and he recorded what the patient's experiences were, as relayed by the person lying on the bed, when he electrically stimulated particular parts of the person's brain. This gave him an opportunity not just to discover where areas involved in language were, and therefore to plan his surgery more carefully, but to explore functions of the brain by this very crude technique. And he gives the most extraordinary graphic accounts of people describing experiences. Sensory experiences, yes, feelings of numbness or tingling in their fingers, funny buzzing sounds apparently in their ears, or visual experiences, including quite complicated things, great swirling shapes, colored shapes in their visual field, and even what seemed almost to be fully rehearsed memories. When he stimulated in the temporal lobe of the brain particularly, some patients gave an account of what they said were past experiences, almost as if they were watching a movie of themselves in the past, and a movie which included not just visual experiences, but the sound and even the smells of previous experiences. One woman described being at a fun fair or a circus where there were animals, and she could smell the animals in her experience, produced by stimulating the cortex. Well, the task of neuroscience, that's a strong enough indication that this lump of matter has some sort of relationship with having experiences, having memories, having thoughts, having intentions, because in some cases patients felt that they wished to move parts of their body. In other cases, by the way, in other places, their bodies moved and they had no idea that was going to happen, and it wasn't associated with a sense of intention. But our job is to give a full account, eventually, of how this lump of stuff can give rise to or participate in all the experiences of human beings, or all their perceptions, their thoughts, their memories, their plans for action, their intentions, and so on. Now, that's a very extravagant claim, even when it's only a claim about an objective. And of course, we're nowhere near that yet. But that has to stand as the goal of what neuroscience is about, particularly this new and burgeoning area within neuroscience called cognitive neuroscience. Let me set the framework for this first lecture, which I call Darwin's Brain, with this picture. This is not a graph of evolutionary time, which some of you might already be deducing from its general form. It's simply an ordinal ranking of different primates, monkeys, apes, and hominids, in order of their relative brain size. What's plotted is something called the encephalization quotient. It's not just straight volume of the brain, it's relative brain size compared with body size, because in all members of any particular group or class, brain size varies with body mass, so that has to be factored out if you want to compare closely related animals. You can see what the symbols mean. The filled circles are different sorts of old world monkeys, the squares are new world monkeys, apes are crosses, and different sorts of hominids are shown by stars, and they're numbered. And generally speaking, the monkeys are down at that end. They have relatively smaller brains. Generally speaking, the apes are sort of in the middle, and the hominids cluster up here. There's a lot of overlap between them. If one were to convert this to evolutionary time, then the members would roughly line up in the same way. There would be some divergences. And what you would see then is roughly a threefold increase in relative brain size through the 20 million, 15 million years of evolutionary time occupied by these different species. The brain has got bigger. And as you'll see a little later, it's got bigger very slowly in ways that might have pleased Darwin, seeking always for progressive changes to explain, in the end, large differences. One symbol stands out from the rest in this function, twice as big as its nearest neighbors, and surprise, surprise, it's us, Homo sapiens. Our brain is relatively bigger. Now, I should say there are professional discussions amongst the people who are interested in the evolution of hominid brain size. There are discussions about how big that difference is, and certainly how rapidly it developed. But the human brain is certainly bigger than that, actually bigger than that of any other species when judged in terms of the number of neurons that it contains. There are elephants, there are some species of whale that have got bigger brains than us. Neanderthal had a slightly bigger brain on average than Homo sapiens. But the neurons are relatively smaller and they're more densely packed in humans. In those terms, we have more cells in our brains than any other existing species. Well, in The Origin of Species, in 1860, Darwin's great treatise, there was no discussion of the brain at all. The only mention of the brain was in relation to his account of the evolution of the bones of the skull, because he saw that the skull was made up of a very large number of individual bone segments, and he traced back the lineage of those bones through different mammals, and indeed beyond mammals downwards to fish, accounting for the general form of the cranium of human beings in terms of its evolutionary origin. Didn't mention brains. Of course, his conclusion in the Origin of Species was summed up by this famous statement about fitness being responsible for adaptation and the environment essentially selecting individuals on the basis of their fitness. So in a nutshell, his idea was that inheritance, whatever it was, he didn't read Mendel as far as we know. He had a copy of Mendel but didn't read it. he certainly didn't know about genes, but he imagined that something must be transmitted by inheritance, and that something must be subject to variation, and the variations must be preserved by inheritance, therefore producing offspring with a variety of different makeups, what we call phenotypes. And the environment acting on those different choices of individuals within a particular species would select the fittest. The fittest would therefore reproduce on average more than the unfit. Therefore, the characteristics, the inheritable characteristics which gave them their fitness would be transmitted. And eventually, if the changes were radical enough, a magical event called speciation was supposed to occur, and a new distinct species was generated. Although he didn't discuss the brain, he certainly did recognize that this general hypothesis would have its critics, in particular when it came to the explanation of very complex structures. And he considered the eye as an archetypical example of the problem. There's a picture of an eye from a textbook, and you'll all know that the eye is a roughly spherical optical instrument, transparent bit at the front, a lens inside which can change its shape and therefore focus the images at different distances. There are actually three coats to the globe of the eye, a mechanical one, a vascular one, and then a neural one, the retina, which contains all these complicated neurons, the rods and cones, but also different classes of cells in between, and finally sends its fibers out to the optic nerve. It's a very, very complicated structure, and it's generated with contributions from different parts of the embryo. The lens, for instance, comes from the ectoderm, moving inwards to join an outgrowth of the brain, which is forming something called the eye cup. Complicated, orchestrated assembly of different parts of the growing embryo to make this perfect, complex structure. Well, Darwin said, setting up a straw man, as it were, in The Origin of Species, to suppose that the eye could have been formed by natural selection seems, I freely confess, absurd in the highest degree. How could tiny little changes from some very primitive sort of eye, even if transmitted by inheritance, collectively produce something as complicated as this? But a few paragraphs later, he quite brilliantly deals with the problem. And this is the remaining classical description of how complicated things in the body could have evolved. Just read it for yourselves. The crucial points are that if each grade of eye, from the simplest to the most complicated, is useful to the organism that possesses it, and if its structure varies just a little from organism to organism in that species, and if the variations could be in themselves be useful in animals under changing environmental circumstances, then you can imagine it's not beyond comprehension. It's not subversive of the theory that there could, over long periods of evolutionary time, have been accumulation of small changes to produce the eye that we have. That view that there might be a very long evolutionary lineage in producing something as complicated as the eye has certainly been vindicated by modern research. It turns out that there is a controlling gene, what's called a transcription factor, and these seem to be crucially involved in regulating how the body develops in the embryo. There's a transcription factor called PAC6, which was first discovered in fruit flies, then a homologous gene, only very slightly different, found in mice, and then in humans. It's in everything. And between fruit flies, or the common ancestor of fruit flies and human beings, well, that's probably, a couple of 100 million years or something like that of evolution. So switching on the production of an eye seems to have been discovered a very long time ago and then conserved, as the geneticist would say, hanging on to some clever little discovery of evolution. So here we are looking in a fruit fly. Here's an scanning electron micrograph of a fruit fly's head showing the ommatidia, the eyes of a fruit fly, incredibly different from ours. Hundreds and hundreds of little facets with a very small group of receptors at the bottom, different sort of optical structure, all clustered together. It looks completely different from a human eye, but it's the same transcription factor that turns on its production in the embryo. Well, in this unfortunate fruit fly, or maybe it's fortunate, an extra little eye is being generated. You can see it here. It's a tiny, perfect little ommatidial eye, in addition to the ones that the fly would normally have. And that's been produced in the embryo by forced expression of that transcription factor, PAX6, in a place where it wouldn't normally be expressed, a bit further forward in the developing face of the embryonic fruit fly. And what it's done is to produce an extra little tiny eye. That's remarkable enough. What's truly extraordinary is that the particular version of PAX6, which had been, as it were, injected into this embryo to make the extra eye, was the PAX6 of a mouse. The PAX6 gene from the mouse will turn on the production of an extra eye in a fruit fly. It's that conserved, the mechanism. It's expressed in human beings. Here's a picture which shows in orange cells within the developing human embryo where this PAX6 gene is being turned on. And as you can see, they're all around this region, which is the developing eye cup. It's controlling it in the human embryo as well. Mutations of this gene will cause disturbances in eye structure, particularly a condition called aniridia, where the iris of the eye fails to form. It was discovered, by the way, in fruit flies through genetic screening natural mutants or mutants produced by X-irradiation, because mutation of this gene in fruit flies stops the eye forming altogether. It produces a mutant called eyeless, and that's how it was discovered. So it's a beautiful example of how a single genetic mechanism can be conserved over hundreds of millions of years of evolution, and how that mechanism can be responsible for producing very different forms of eyes, depending on all the associated selection that's going on after the initial switching on of the eye production. Well, what about brains? Brains have increased in size, perhaps very dramatically in relatively recent hominid history, in us. Can we really imagine, even the processes that explain the complexity of the eye, can we really imagine that similar things have produced the human brain? Well, first of all, it's got to get bigger. Secondly, the bigness has of itself got to be useful in some way that's selective. Well, it turns out that getting bigger is no problem. If you think about it, the problem is actually controlling the size of anything in a growing animal. Why are we, 5 or 6 feet tall and mice a few inches long? What is it that determines the size and the adapted size, the optimal size of a particular organism? Well, it depends how you ask the question why, or how in a Darwinian sense must have something to do with a particular environmental niche for which it's been selected, for which we've been selected and mice have been selected. But in terms of mechanism, it's more difficult to understand. We and mice are produced by the proliferation of stem cells. We start as a single cell, and then it divides, and lots of them. Then they start to get committed to different organs, and it goes on growing until the stem cells stop. And then thing stops growing and it becomes of its full size. So size is determined by how quickly and how often stem cells divide. If you think about it, it might not be too difficult to make a brain which is bigger, say twice the size. All you need is, on average, 1 extra cell division in size. Or if you have 2 extra cell divisions, it increases by 4. So in principle, it might be very, very easy for evolution to have tinkered with the control mechanisms for stem cell division to produce a larger brain. And you can do that. Right? By you, I mean some clever molecular geneticist mutating mice. There are 2 ways that have been described to do it. You can change a gene that disposes cells to die. Cells die, lots of cells die during normal development. So if you can reduce the number of cells that die, then things will more of them will survive, and therefore the things will get bigger. So here's a mutation in mice that affects a gene that determines the probability of cell death. And here are normal mice embryos, here are the mice embryos, and these are embryos with the mutation. And what you see is that the forebrain swells up hugely in this particular mouse, simply because fewer cells are dying than normal. Even more dramatic is the other way of doing it, which is to change the proportion of the stem cells that re-enter the cycle. In other words, carry on dividing. So the divisions just continue longer. And this mouse survives, it's born, its head's a bit bigger than normal. If you look inside its head, you find this extraordinary brain. So a cross-section through the brain of such a mouse, and what you see immediately is that it's folded. It's got big, deep folds in it. It has to, because there's so much brain, it can't cram into the skull of the mouse without folding up, which is exactly what the human embryonic brain does during development. Because it's growing so quickly, it's constrained by the skull, and it folds to form the characteristic folds of the brain. It can happen in a mouse. It never does in normal mice. But this almost looks like human cerebral hemispheres, just produced by tinkering with the cycle. It's very easy then to make a large brain. In fact, you might then ask, and Darwin did, why it is that brains haven't got bigger amongst all types of animals. Why don't we find mice with huge brains? Wouldn't there be some advantage from that? We've got big brains, must be an advantage. I'll come to that. So why not mice as well? Darwin talked about variation even between insects in the sizes of their brains and related it to their behavior, and claimed that insects with slightly bigger, tiny, but slightly bigger than other insects were more intelligent. So he suggested that there was a relationship between brain size and intelligence. So why hasn't every species taken advantage of that in order to have some evolutionary advantage? We'll come to that question in a minute. Part of the question's answered here. I just want to give you, to set the context, not just for this lecture, for others, some numbers to hold in your heads about the scale of the problem that neuroscience is trying to solve. And, okay, that's partly an explanation for how little progress you might think we've made so far, but it's also an indication of how much potential there is for explanation of very complicated things in our brains. The human brain has about 10 to the 11 neurons. That's a very large number. That's equal to approximately to the number of bodies in our galaxy. But the big numbers start when you consider that every one of those neurons, on average, is contacted by about 10,000 other neurons. So it's the connectivity which is interesting. So if there are 10,000, 10 to the 4 connections onto each neuron, the 10 to the 11 neurons, it means there are 10 to the 15, one with 15 noughts after it, connections. And now that is a very, very big number by anybody's standards. Just to set it in context, the lifespan of a human being is, if you work it out in seconds rather than conventional years or decades, is about 10 to the 9 seconds. So if we have 10 to the 15 neurons and we live for 10 to the 9 seconds, then on average through our life, we're making 10 to the 6 connections per second. That's 1 million connections per second. Now, I just paused for a moment, not just for dramatic effect, but to let you make a couple of million connections so you might remember what I said. Because certainly the breaking down the formation of connections, although much of it happens in the early years of life, is going on through the whole of our lives. If I can cut to the chase, that will be one of my conclusions from this lecture. The brain is constantly changing, and some of those changes are certainly involved in laying down memories, in learning, in what's come generally to be called plasticity of the brain. The brain, though, and this is now tackling the question of how it evolved. The brain is metabolically very greedy. Your brains are consuming about 20% of the oxygen and glucose in your blood. it takes a lot of energy intake to maintain a big brain, and this is probably the primary constraint on the evolution of big brains. Brains are kept by evolution as small as is possible to do the job they have to do, and you see all kinds of specializations within brains of different species that indicate that. Unnecessary bits become relatively smaller, essential bits are conserved or evolve to become relatively larger, but brains altogether are kept as small as they can. Well, that makes the enlargement of the brain in hominids even more interesting. What allowed it to escape from this constraint? What could have been the huge advantage of having a big brain that enabled us to deal with these horrifying demands that the brain presents for oxygen and particularly for food, for nutrients? Just as an aside, an awful lot of our brains are devoted to vision, and I'll show you some examples of that and in other lectures as well. Probably, by my estimates, about 20 billion neurons in your head are primarily devoted or exclusively devoted to visual analysis. So that means that seeing takes about 4% of everything you eat. So if you're worried about your weight, don't pay attention to all these slimming regimes that you could pay a lot of money for, go to health farms or whatever. Just go to a good movie and let your visual system burn it off. Darwin didn't know much about metabolic demands of the brain, and as I said in the first edition of The Origin, he never mentioned the brain at all, at least not in evolutionary terms. It was just something that was inside an interesting skull. But let's just think about what he did. What was the historic context in which Darwin was thinking at all? Don't forget that he basically developed his ideas about natural selection very much earlier than The Origin. Certainly by the late 1830s, his views were pretty well formed after the voyage of the Beagle. At that stage, at least popular thinking in Europe about brain function and the relationship between brain and mind was still dominated very much by the ideas of this man. Amongst neurologists and even most psychologists, his views had fallen out of favour, but they're still very popular in the salons of Europe and Victorian England. This is Franz Josef Gall, an anatomist and clinician who worked in Vienna at the latter part of the 18th century into the beginning of the 19th century, and he observed the structure of the human brain. He was interested in the organisation of the human brain. He dissected from cadavers to look at the general form of the brain, but developed the view that it was possible to diagnose something about the anatomical structure of the brain without dissection, simply by feeling the shape of the skull. That turns out not to be at all reliable as an indicator of brain form. Many parts of the brain aren't directly in contact with the skull, and even where they are, there's not an obvious relationship between the bumps on the head that Gall was describing and the relative size of structures of the brain underneath. But fundamentally, his idea was that if we could look at bits of the brain, diagnose the relative size, we might be able to relate that to the personality, the characteristics, the abilities of the person, the mental abilities of the person. So pictures like this became very popular. This is, of course, phrenology. There was a phrenological society in Philadelphia until the late 1950s. So the ideas persisted for a long time. And certainly in Darwin's time, they were still talked about a lot in polite society. Interesting comment on the way in which the social context sets the kinds of questions that scientists ask. Because what did Gall expect to be localized in the brain? Things like secretiveness, sublimity, cautiousness, self-esteem, firmness, good, strong, robust Victorian characteristics which distinguish one person from another. Nothing about vision, movement, hearing, or touch. There was an American version of this at the beginning of the 19th century that had republicanism as one of the essential organs. He was very much criticized quite soon after announcing his ideas. I mean, there were very serious scientific articles. His colleague Spurzheim wrote a series of papers, long papers in the Lancet, first paper in the Lancet week after week describing this in the early 19th century, but it sort of fell out of favor because people recognized the poverty of the number of observations, the lack of controls, for instance. Could you find people with the same bumps who hadn't got the things that he said were attributed to them and so on? So it kind of fell apart very rapidly as a theory, but it had a kind of attraction to it. Because it fell apart as in a theory amongst serious neurologists and scientists, it actually held back significantly the now widely accepted view that functions are localized in the brain. Not these kinds of functions, but others, as we'll see in a moment. But Darwin knew about this and refers to phrenology quite frequently, actually. Interestingly, if you compare Goll with phrenological, he has a very interesting bump at the back, which I think probably most closely corresponds to the organ of self-esteem. That's not surprising. Darwin also knew about the work of this man and referred to it. Extraordinary British neurologist David Ferrier, working in the West Riding lunatic asylum, which might sound an unlikely place for a serious academic to be working. In fact, it had been a centre of serious research for a long time. Even more extraordinary were the things that he was doing in the asylum in the West Riding. He was performing really quite sophisticated physiological experiments on the brains of apes, of gorillas, chimpanzees, orangutans. Now, the thought of how you get an orangutan into a lunatic asylum in the West Riding blows the mind, really, but it happened, and he anesthetized animals, stimulated the surface of the cortex, the same kind of way that Penfield used 100 years later, and derived from that maps, diagrams showing localization of areas of the brain. This is the brain probably of a chimpanzee, actually, showing regions where stimulation produced different sorts of movements of the digits, of the lips, of whole arms or parts of the body, of the eyes, and so on. And he related that to localization of function and post-origin of the species extrapolated to human beings. This is a human brain, and he imagines the same localization, equivalent localizations are in humans. Darwin certainly knew about his work after publishing The Origin and referred to it. Well, what exactly then is the relationship between brain size and adaptation of value in Darwinian terms? Darwin discussed in a later edition of The Origin, when he just started to talk about the brain, this issue of why it was that brains didn't increase in size in different species. Couldn't come to a conclusion, but he asks this interesting question. No one can solve the problem why of two races of savages, two different strains of human beings, two different isolated groups of human beings, one has risen, as he says, higher in the scale of civilization than the other. And this apparently implies increased brain power. So he's saying that even if you compare amongst human groups separated from each other, some might be more advanced than others, surely this must have to do with the differences in brain power. He doesn't say brain size, but brain power. And that, of course, is the key to the way that we think about the relationship between brain size and brain function, and the key to what, as far as I can see, every paleoanthropologist thinks about the Darwinian interpretation of the evolution of brains. That big brains are clever brains. Having a big brain makes you clever. So here we are, shown very clearly in this magazine. This is your brain before you read the magazine, and it makes you smarter but it makes you smarter by making your brain bigger. So big brains are clever brains. But what do we mean by clever? If there's a relationship between cleverness and the size of the brain, cleverness, knowledge, if you like, obviously involves much more than just having a structure. It requires putting into that structure information. And information of a particular, which I'll talk about perhaps in the second lecture, information of a particularly interesting form, because the information that our brains contain allow us to understand each other and understand the world. It's not, as it were, robotic information. It comes with implications about meaning. If you like, it's semantic. So that surely isn't possible without knowledge and without experience to give you knowledge. So having a big brain isn't enough. You need to use it to gather information in order to acquire knowledge. All right, but the idea basically is that human beings have this capacity to using their big brain to reflect on the past, to plan for the future, to consider different scenarios. That's what makes us clever, and that was the biological advantage that conserved our otherwise disadvantageous big brain. It doesn't always work. This is a picture I plucked off the web, which shows these guys who've been spending a busy day installing all these very robust metal posts here in order to stop people parking next to the building. So they knew what they were doing, but didn't reflect too well on the consequences of their own actions. Okay, so let's just step back then and ask how is the brain organized in a way that might allow it to capture and express knowledge? Well, in Darwin's time, neurologists already knew that the human brain, this is a lovely picture by Gratiolet from the beginning of the 19th century, the human brain with all its folds, like the mouse, remember, with all these folds, enormous area of cerebral cortex, because this is what the business end of the brain as far as cognition is concerned, the cerebral hemispheres, does have localized functions. They weren't the kinds of things that the phrenologists talked about. They're much more kind of routine, mechanical sorts of things. So there's a strip in the precentral, what's called the precentral gyrus there, which is responsible for the most direct control of movement. If Penfield stimulated there, the patient would see their fingers moving. They didn't have any sense that they were causing the movement. It was quite a surprise to them. And as they moved the electrode along, different parts of the body would react on the opposite side of the body from where he was stimulating. Very kind of regular arrangement, the representation of the body feeding out into the spinal cord to control the muscles. Immediately behind that is a strip that receives information from, it's a sensory area, receiving information from receptors in the skin, the deep tissues of the body, which again has a kind of representation of the body, the feet are at the top, the head's towards the bottom, heads and hands, and that is lined up with the motor area and the connections between them. It's receiving information from the receptors in the skin. There's a visual area at the back, right back here, most of it tucked inside in human beings. Damage there can cause blindness. Even if the eyes are completely normal and functional, an injury here, a massive stroke at the back can cause a cortical blindness. And then a region here at the top of the temporal lobe, which receives information from the ears. So there it is. As I said, very different areas from those that Gall had imagined, but from a sort of materialistic point of view, makes sense. Information comes in from the sense organs, gets kind of processed and then sent to control movements. But what's all this stuff doing in between? I think this sort of picture, which became quite familiar in popular culture, the end of the last century, the beginning of this century, I suspect that's what led to this very common view that we're using only about 10% of our brains, and if somehow we could bring into action all the rest of this stuff, would be very much cleverer. It was sometimes even called silent cortex, the beginning of the 20th century, or more commonly association cortex, implying that it's sort of associating things that are coming together from the sense organs and then processing them and then feeding them forward to control movements and actions. If we look at in more detail at the way in which the brain has got bigger during evolution, in relation to these specialized areas, it's really quite interesting. So this picture summarizes, 150 million years of evolution, 120 million years maybe, in a single diagram. What it's doing is to show the organization of the hemispheres. These are all pictures of the cerebral hemispheres of different existing species, which are thought to be pretty representative of common ancestors. This is the evolutionary tree here going up towards human beings at the top. So if we look, these are marsupials down here. If you look at placental mammals, then hedgehogs are probably best living representative of the earliest forms of placental mammals after the age of the dinosaurs, the early insectivores. So here's a hedgehog brain. It's tiny. The scales are not accurate. The hedgehog's brain is very much smaller than the human brain, and this implies. But there's a hedgehog brain. The different colours show the different sensory areas of the brain. The touch, the somatic areas, the auditory area, and a couple of visual areas. And what you see is that they're generally laid out across the hemisphere. This is the front, that's the back. Here's the touch stuff, here's the auditory one, here's the visual at the back. And that's true in all species, even by the way down here in marsupials. They all have the same general arrangement. But you can't get from that to that just by blowing it up. It's not as though it just being pumped up like a balloon. All the areas are there in humans, these basic sensory areas, but there's much, much more in between. So the addition of extra brain hasn't been just a kind of uniform process. It specifically occurred between the major sensory areas, adding more and more association cortex in between, which implies that it's the association cortex that's been, as it were, adaptively valuable. All right, what do these areas actually do? If we think about not just pictures with colours on them, and of course there are sophisticated ways of looking at pictures of brains with colours on them as well. The whole modern burgeoning field of neuroimaging, based on brain scanning, shows lovely images of human brains with blobs of colour on them. And there is a temptation to think in terms of the unit of functioning of the brain as being a coloured blob. You must never forget, and I'm not implying that at least the more sophisticated users of neuroimaging do forget, but never forget that one of those blobs in a pretty neuroimaging picture represents tens of thousands, maybe millions of individual interconnected neurons, and it is those neurons which are performing the computations that underpin whatever the brain does. So what do these blobs contain? Well, they contain neurons. And those neurons are capable of quite extraordinary and sophisticated expressions of their own knowledge of what's going on. I'm not implying that neurons have understanding, have perception, have consciousness, but they contain the signals that are necessary for the interpretation and the understanding of sensory inputs. This whole area was very much stimulated by the work of these two people, David Hubel and Torsten Wiesel, who won the Nobel Prize in 1981, I think, for their work on the visual parts of the cerebral cortex. I mean, already a fair amount was known about the eye and the pathway up to the cortex, but the properties of nerve cells in the eye are relatively dull compared with what happens in the cortex. As you might expect, the photoreceptors of the eye respond to light. They respond in peculiar ways, but they respond when a light goes on them. And that information is passed backwards through cells that essentially pixelate the image like a digital camera. So they describe the brightness or the darkness of spots on the retina. So they're basically representing the image to the brain in terms of the lightness and darkness of an array of pixels across the retina. But suddenly at the cortex, the cerebral cortex, an extraordinary transformation takes place. And it's shown here. This is one of the early illustrations in a paper by Hubel and Wiesel with the techniques of the day, in the 1960s, photographing directly from an oscilloscope screen connected to an amplifier into which was feeding a tiny, tiny microelectrode that had been introduced into the brain in an anaesthetized animal, cats, later on monkeys, to pick up the impulses, the little tiny voltage impulses, from an individual nerve cell. And that's what's shown here. These little blips are the clicks produced by the firing of one nerve cell that we're recording from. What you see, and this shows time, this is time, so suddenly the cell goes, it stops. It's silent, it doesn't go here and here when there's nothing happening in the visual field. The lights are on, but there's nothing specific happening. When this pattern was moved across in front of the eyes in a particular position on the screen in front of the animal's eyes, the animal's lying there anaesthetized, as it moves across, when it hits a particular point on the screen, in other words, a particular spot on the retina, the cell gives a big burst of impulses. That's nice. That's what you'd find if you were recording from a cell in the retina as well. It would just respond to light or to darkness in a particular place. What's special about the cortical cell is this. They changed the angle of this bar on the screen, moving it across the same position of the screen, and the cell stopped responding if the angle was changed. It didn't respond to a horizontal line or a vertical line. This particular cell responded to an oblique line. Different cells respond to different lines. They're beginning to, if you like, to pass the image. They're beginning to break down the boundaries of images of objects into their component orientations. That's the sort of dogma. And then sending that information on to the rest of the brain to help to understand what's out there. So this was a remarkable finding which galvanized the field, because here were neurons, capturing a bit of empirical information about the nature of things in the world. The cortex is beginning to describe the world rather than just, as it were, reflect the world. Well, it turns out that their recordings were taken from the very first classical visual area known to the 19th century neurologists. But when they and others started to apply their attention and their microelectrodes to the surrounding association cortex, the things that had been added during evolutionary development, apparently silent, they found that they were not at all silent, that they were also packed with neurons that responded mainly to sensory stimulation. So around the visual area is an array of other visual areas. This is 1980s picture, but it sort of sums up the state of knowledge at that stage. And it describes the old world monkey brain. This is a picture of the brain of a rhesus monkey, viewed from the side. Here's the front, there's the back. The primary visual cortex is that big red blob at the back, more exposed than in the human. In the human, as I said, it's tucked more in. There it is. That's the region the human and Wiesel had studied. But here, in this picture, this is an imaginary flattened out view of the cerebral hemispheres, as if they'd been peeled like the skin of an orange off the surface of the cortex, then flattened out. These were all the different areas that had been described by electrophysiologists, recording in different places. And all of these in the back of the monkey brain, stretching down the temporal lobe up into the parietal lobe. All of these things with V's on them, but many of the others as well, were all visual. The cells in them responded to visual stimulation. Well, where did they get their information from? Not from the eyes directly. The information from the eyes comes almost exclusively into the first visual area. And that's true in hedgehogs as much as it is in humans. But there's a complex array of interconnections out of that first area into all these other areas, sending information on for, as it were, further analysis. And it turns out that, and I should say that many of the first observations in this field were done in London by Semir Zeki at University College, who was the first in monkeys and then extending into humans to suggest that these different extra visual areas might be committed in some way to an aspect of interpretation, some particular aspect of interpretation of the visual image. He was particularly interested in two regions, this one called V4 and the neighbouring region called MT, which he calls V5. Although they're adjacent to each other in the monkey, in any case, they seem to be committed to very different roles. So there's some degree of specialization within these regions. MT, this region MT has been, just digress for a moment because it's worth saying what kind of specialization we're talking about, remembering that these things might have been added during evolution to give animals an advantage over those that only had the early visual areas, not the later ones. MT contains neurons which respond to moving images. Now that's not surprising. Many of the cells in V1 also respond to movement, and indeed some of them even respond selectively to movement in one direction rather than the opposite direction, so-called direction selective. Well, all the cells in here are direction selective. And pretty much none of them is selective for colour. They just receive from all the cones together. They're not interested in colour, whereas at least some of the cells in the neighbouring V4 area are highly sensitive to colours, different cells responding to different colours. Well, you might say if cells in V1 respond to movement, and they send information to MT, which they do, and the cells here respond to movement, what's being added? Well, that was first looked at by Tony Movshan in New York, Bill Newsom at Stanford, Bob Wirtz at NIH, who described remarkable characteristics of cells in that area which are not present in the cells that provide their input, which is a little bit surprising. And I'll show you the sorts of stimuli that they used. They used patterns like this, so-called grating patterns. Now, if you move a pattern like this across the correct bit of the visual field while recording from a cell in the primary visual cortex, which Hubel and Wiesel described, then yes, the cell responds when the angle of the lines is correct, and if it's direction selective, when the direction of movement is correct. And if you rotate the pattern, it'll stop responding if you move away from the optimum. Now, it's a real pity I can't show the demonstration. If you combine two of these things to make what Tony Movshan called a plaid pattern, and you simply move both of them, that one and this one, you combine them together, what you see, and it's an overwhelming, irresistible visual illusion, if you like, is a sense of movement in the shared vector of motion, the average of the two directions of motion. And this is an essential part of the vision of moving objects. If you think about it, if you look at an object like my hand moving around in the field, and you want to know where my hand is moving, then you could analyze the various edges and boundaries within my hand and at the edges of my hand, and then track them. But if your tracking was based entirely on, was tied to the particular orientation of movement, then you couldn't tell the difference between, well, my hand moving in that direction, for instance, or my hand moving in that direction. This edge of my finger would always be translating in that direction, as far as you were concerned, if you couldn't take into account all the other motions that were happening at the same time. So looking at all of the component orientations and their vectors of motion and resolving them all will give you the single shared vector, which is the actual direction of movement of the hand. Now, it turns out that the cells in MT, or many of them, solve this problem. They can do the computation, which allows them to combine information from different angles, and they will respond either to a single line or a grating pattern like this, or to the sum of gratings if the whatever the perceived direction of motion is corresponds to their preferred direction of motion. So the output, if you like, if someone were looking at, if a little homunculus were in there looking at the output of a cell in MT, it could tell from the activity whether the motion of the single object or the surface was in a particular direction. Cells in V1 can't do that. It can be derived by looking at the spatial and the temporal correlations of activity within V1, detecting information which is unknown to the individual cells in the first visual area. So these extra areas, if I can, incredible sweep of generalization, these extra areas seem to be looking progressively at the undetected spatial and temporal correlations of activity amongst the neurons that are providing their input. And then, as the jargon goes, making that information explicit. The information exists, but it can't be recognized by the individual neurons. When it's combined, it's made explicit in the properties of the neurons that detect it. Well, I mentioned neuroimaging, which has added hugely to our understanding of two things. The maintenance or conservation of properties between animals and humans, and the characteristics of the brain that are thought to be very special, maybe even unique in humans, such as a localization of language functions. So this is one of the scanners in Oxford, an MRI scanner. And I speak with trepidation because I know there are real experts on this in the room. The MRI scanner essentially measures changes in local blood flow in the brain, at least what's called functional magnetic resonance imaging does. You can look at blood flow in the brain and produce a statistical description of changes in blood flow in relation to things that the person is doing when they're in the scanner, looking at things, listening to things, moving their fingers, or whatever. So you can ask which part of the brain had a statistically detectable change in blood flow in association with some particular thing that the person was doing or thinking about or listening to or whatever. So to give a very simple example, if you put someone in the scanner, measure the general flow of blood in their brain using this technique when they're resting or when they're looking at a blank screen, and then you have a screen in front of them and you show a pattern like this, luminances, brightnesses and contrasts, and ask what part of the brain increases its blood flow compared with the resting state. For that, what you see is this with the red blob again, red blob at the back of the brain. This is the front of the brain, that's the back of the brain, in and around the classical visual areas. What you'd expect, the person sees it, their visual cortex has been activated by their eyes, so it lights up. But what about if you then show them this? Again, a pattern of dots, but it's moving around, and you ask, what part of the brain is active, it has its blood flow increased by this, over and above that. So you take the difference between this activity and that in a so-called block design, and what you see is this. This is this classic area of MT, or V5, the motion selective area, in humans, which we presume has the same kind of nerve cells that monkeys do, doing these clever things to compute true movement in space. Now this pattern is not, this is really important because it's a very common misunderstanding of these sorts of things. This pattern is not the activity, this is not the activity produced by that pattern. That also activates these areas, and there's all kinds of statistical thresholding as well to give a nice clean picture. This is the additional area of blood flow generated by that compared with that. So this is activated sense things. So here they do more stuff than they're activated by the movement. And here is one of the early pictures showing a rendered brain. It's much more sophisticated these days, but I think this is one of Semiozeki's early pictures of a human brain, right hemisphere, left hemisphere. And the red blob actually is the result of a different technique called positron electron tomography, showing the same region, the V5 or MT, activated by movement, just here, right in the middle of your association cortex. So about a third probably of your cerebral cortex is devoted to vision, and it consists of lots and lots of different modules doing different sorts of computations. So for instance, if you stick someone in this is an experiment done by Tim Andrews in my lab, but repeating what had already been done by others, you show people a series of photographs of real faces. And you interleave those photographs, 10 seconds each, you interleave them with photographs of man-made objects. So the person just lying there, face, object, object, object. And you say to the computer, right, show me the parts of the brain which responded more to these than to those, and vice versa. And you get nice pictures, which in a hand-waving way, and a wrong way, might encourage you to say, well, you're looking at a face processing area or an object processing area. This is one such image. So here's a section, human brain, this is the right side, that's the left side. Lower down, you can see the eyes there, it's lower down. This is actually the top of the temporal lobe, leading into the temporal lobe. And what you see is this orange blob here, which was the region which was more activated by the face images than by the objects, and as it happens, a nearby region, small region, which was more activated by objects than by faces. I won't go, perhaps I will more in a future lecture, but I won't go into the evidence for why this is most certainly not an object processing perceptual area, which is telling you about objects. But this one, I think by a lot of, an enormous amount of evidence from different directions, does seem to be in the mainstream of telling you that, something about faces. So the notion is then that there are a large number of modules, some of them only really fully developed on one side of the brain, which are processing different aspects of the visual scene. There are others around the auditory cortex, there are others around the somatic sensory cortex, and a whole slew of stuff at the front, which is helping you to plan your actions, make decisions, and so on. The whole of the cortex is laid out in these computational modules. There is no emptiness, there is no silent cortex, there is no association cortex. It's all doing stuff. It's all processing and computing away, and that's been added during evolution. Well, how? How do you get, a module generated with hundreds of thousands or millions of neurons wired up so they're able to do some clever computation like this direction of motion thing by a Darwinian process? Getting extra brain is easy. Getting extra brain that's doing something useful is much more difficult to understand. Well, let's get a kind of hint of it. This is a study published fairly recently, looking at the result. This is from fMRI, looking at the activity in the brain, if you'll accept shorthand, for 3 different stimuli. The one produced blue, the next one produced green, the next one produced violet. The yellow is the overlap between the green, not a different stimulus. Right, 3 different stimuli. What were those 3 stimuli? Well, they're at least some of them are visual. We know that at the back of the brain here, this is the primary visual cortex, it responds to static contrasts, just any sort of visual stimulus. This activity was produced by moving stimuli of different forms. Gosh, my computer doesn't seem to like this arrangement anyway, but if the stimulus is moving, then it really doesn't. That's very interesting. Okay, so this should be moving back. Oh my, it's going to ruin my next. That's a shame. My next demonstration is going to work. Anyway, this then is produced by motion. This is relatively more activated by moving stimuli. This is MT. This is probably V2 actually, which is also more active than V1 for motion. That's fine. What produces the green area? What was the stimulus for that? Well, unfortunately, I don't think this is going to work. Oh, it is? Splendid. Great. I'm glad this movie works. So the stimulus in this case was that. Also a collection of little moving dots. But I think you'll agree, you immediately see that as representing a moving figure. It's computer generated. What it represents is the relative motion of the points of articulation of the body. You instantly see it as a moving figure. And we are definitely specifically tuned to see that kind of visual stimulus. There's lots of evidence that we handle that by a different process, neural process, compared with just recognizing linear motion. And indeed, there's an extra bit of brain right here in the association cortex, as it were being tagged on to the mainstream of visual processing just to do that sort of job. Right, what would you think, anyone who hasn't read the paper, was the stimulus that produced this big lilac bulb? We're going from static patterns to ordinary linear motion to interesting forms of non-biological motion through to this thing called biological motion. What's the next one? Anyone want to hazard a guess? Self-motion. Actually, there are bits down here that respond to self-motion and mouth movements and things, all sorts of interesting things. No? Put you out of your verbs. Verbs. Spoken or written verbs. No, again, a lot of hand-waving, sort of relationship between motion and its analysis and expression through verbs. Not all verbs, of course, are verbs of motion, but you might imagine that the first verbs that humans employed, go, pull, stab, probably were a lot to do with movement. So it could be that the bit of brain which becomes occupied by representing that part of the lexicon is associated with a stream which has evolved for the analysis of movement. There are other regions, by the way, that respond more to things like adjectives and nouns, which seem to be further down and closer to regions that are analysing objects and forms. So, again, it's not a theory. It's not a theory, but you could imagine as bits of brain have been added through a very simple process of mutational change, just generating an extra bit of brain. They might, in each individual, have become occupied by connectivity, which enabled those neurons to do bits of correlation and analysis on their input. And what kinds of computation was done would be determined by what was there already. So the general arrangement, the similarity of the arrangement amongst individuals might almost be fortuitous, just depending on what is there to begin with. And indeed, you can stand back from that and say, well, how much of this then really is predetermined, and how much of it depends on the individual experiences of this person? This area for verbs, well, no one knows verbs when they're born. I mean, everybody has to learn the lexicon of verbs. So, all right, this region is ideally placed, and even one might say committed to doing verbs, but it doesn't know anything at birth. It has to be exposed to verbs, and the very particular verbs that you know before its knowledge can be captured. So what about MT? Well, very recent work that Tony Movshan's been doing shows that in monkeys, cells of MT respond to moving stimuli very early in life, within the first few days after birth, and they are selective to the direction of motion. But they're not yet doing the clever things that are characteristic of MT in the adult animal. They can't solve this computational problem of recognizing the movement of two grating patterns added together. They respond to only one of them individually. They don't solve what's called the aperture problem in computational vision. And that takes months and months and months to acquire, presumably through the experience of the animal. It sees lots of hands moving, sees things moving around, and recognizes from the conjunction of input from V1 what a moving object, what sorts of activity a moving object has associated with it. So these, although the locations are in a sense genetically determined, the knowledge might not be. So here we are then, that area that responding to verbs is bang in the middle of Wernicke's area. This is the left hemisphere, that was the right, but we know that initially there's pretty much symmetry in the language areas. This is the region which, when it's damaged in adults, on the left side anyway, causes a very strange aphasia in which people can't understand what's being said to them. They can't, and they become deeply dyslexic as well. They can speak, and they speak, as it were, grammatically, but it's completely not what they say is nonsense. Whereas classical damage in Broca's area there produces the opposite, almost the opposite syndrome, in which the patients can understand what's said to them, they can read, but they simply can't speak, they can't find words, they certainly can't put them together grammatically. This is a complicated, I'm getting close to the end of the story. It's a complicated picture, and I'll try to explain it in a simple form, so you're just going to get it from the images. This is a study, it's got blobs on it, of course, but it's not fMRI. This is not showing activity, it's not showing blood flow. It's showing relative thickness of the grey matter of the cortex, expressed in terms of colour. So it's using a technique called voxel-based morphometry, just looking at the structure of the brain, using MRI to look at brain structure. And it is comparing the relative thickness of the grey matter between pairs of individuals. Each of these columns represents a comparison between one set of pairs of individuals, where the colour describes how similar the grey matter thickness was amongst the two members of that pair. There are lots of pairs that contribute to each. There's a big sample of pairs in each of these columns, 3 different categories of pairs. These are unrelated people of the same gender and the same age being compared. And you say, well, they're all blue. They're all apparently exactly the same. Well, that's just a statistical manoeuvre. They're being normalized. So the degree of variability that you find between unrelated subjects is expressed by that blue colour. And any deviation from the blue down towards red shows more similarity between the particular people who are being compared than between totally unrelated pairs of people. Okay, well, these are interesting comparisons. These are fraternal twins, not identical, and these are genetically identical twins. And you see much more red and stuff here, indicating that they're much more similar in grey matter thickness at any point in the cortex than are unrelated people. Surprise, surprise. So the genes are constraining grey matter thickness. But where are they constraining it? Well, where they're not constraining, even in identical twins, there are regions which are as different between pairs of twins as between pairs of unrelated individuals. There are other areas which are much more similar. They tend to be the primary areas, the motor and sensory areas here, visual, the back here, auditory cortex. But a lot of the association cortex is, as it were, individually variable, even between identical twins. So there's a strong genetic constraint on grey matter thickness, but it's not complete, and it tends to be less complete in later evolved regions of the cortex. Back to Darwin. Darwin had a curious episode after returning from the voyage of the Beagle, and maybe very significantly before proposing to his cousin. Remember, this is the time he went through the exercise of writing in his notebook the advantages of marriage and the disadvantages of marriage, and listed them all. Advantage was, someone to look after me when I'm old. Disadvantage was, it'll interfere with my work. And since the column of advantages was slightly longer than the column of disadvantages, he decided to propose. It was an odd period. He went back to Scotland, where he had been a medical student for a brief period of time, but he didn't like the surgery. But he'd been a medical student there. And he began to write in a number of notebooks. This is 1838, so this is, his views about evolution were pretty much formed, but a long time before The Origin was published. One of his notebooks was, as he called it, his metaphysics notebook, his M notebook, July 1838. And he wrote these things. One is tempted to believe phrenologists, he knew about phrenology, are right about habitual exercise of the mind altering the form of the head. And that's a feature of phrenology they haven't mentioned so far, and it's now critical. Garland-Spurzheim suggested that in that good Victorian way, anybody could rise, to improve their character through education and so on. And since character was correlated with brain size, it must mean the brain is capable of growing, changing, as a result of personal effort and activity. And here is Darwin reflecting that. Those qualities, he then says, become hereditary. Well, isn't this heresy? Isn't this anti-Darwinian? How can something that you acquire through exercising your mind and therefore the form of your head become hereditary? When a man says, I will improve my powers of imagination, and does so, is this not free will? But he's saying free will leads to changes which then become hereditary. Well, he didn't, of course, know about genes. He didn't know about inheritance, but he was certainly implying that the mental characteristics could be acquired. I'm close to my conclusions. And again, to avoid stating how far I believe in materialism, this was a real tussle he was going through at the moment, the extent to which he should publicly declare his own commitment to materialism. Saying only that emotions, instincts, degrees of talent, which are hereditary, are so because the brain of a child resembles parent stock. And phrenologists state that the brain alters. So it's a very strange Lamarckian sort of view, and actually much of his writing, especially in The Descent of Man and the expression of the emotions in man and animals, was very close to the bone. I mean, it was very Lamarckian in view. Forgivable because he didn't know about the units of it. He didn't actually know how inheritance works. Finally, then, this is the question that we really face in trying to give an account of how the human brain evolved. And I quote from my good friend Stephen Oppenheimer, who's a geneticist and a real expert in this area, the idea that we somehow grew a big brain first and then decided what it was for is a negation of the principle of natural selection. New kinds of behavior precede the physical adaptation that evolves to better exploit that behavior. This is the dogma. So it must be saying that something must have happened in the environment, as it were, which made it an advantage to have a bigger brain, so allowed the brain to evolve into an environmental niche. And if brains are all about being bright, then it must have been the demand for brightness, the opportunity for brightness to make a difference that encouraged the increase in brain size. Well, this is an illustration from Oppenheimer's book. And it shows now in evolutionary time, the last 2.5 million years. And this is Oppenheimer's interpretation of brain size, by the way. It's a bit different because, so here we are. This is his version of how the brain has increased in size. A bit different from the one I showed earlier because this is absolute brain size, not encephalization quotient. But anyway, it gets very, very big, and it reaches its peak by 200,000 years ago, which is when the speciation of Homo sapiens is supposed to have occurred. But on the same graph, this is his estimate of the evolution of cultural innovation. So we've got stone tools. Stone tools were used by pre-sapiens hominids, but they gradually became more sophisticated. This is shell fishing, long-distance exchange, the beginning of cultural exchange and trading, presumably, exploration, fishing. What have we got? Microliths here, metal, direction of metal, writing, reading and writing 5,000 years ago, musical notation, and going to the moon. That's science and all that kind of stuff in the last 500 years. I mean, there have been no additional genes, certainly, and really very little genetic change in the last 200,000. Odd little polymorphic variations, getting whiter as you go further north and things like that, changes in hair color, differences in size and stature. But what's this? The last 500 years, there's this explosive increase in, if you like, cleverness. But isn't that what big brains are supposed to be about? Isn't that supposed to be the thing that drove the big brains? It was the cleverness, the capacity. So I talk endlessly to, and I'm no expert, but I talk to paleoanthropologists about this, and they say, well, yes, but there must have been, little advantages. And you say, well, what's the evidence in the fossil record? As far as we know, for 50,000 years, early Homo sapiens lived in ways, style of living, community groups, style of foraging, and so on, indistinguishable from modern chimpanzees. And they did that with this massive, hungry brain on their heads. Huge Darwinian disadvantage, no obvious comparable advantage. That comparable advantage is appearing now, and who knows, when it's going to stop. I'm not going to explain, I have ideas, but I'm not going to explain it. There's a huge gulf here between the facts of anatomical evolution and genetics as well, all of which fits perfectly with speciation, essential speciation occurring 200,000 years ago or more, and then followed tens, even hundreds of thousands of years afterwards by the discovery of what this thing in our heads is capable of, cultural evolution. Well, I want to finish by, yes, I must finish, by going back. As far as I know, Darwin never refers to Willis. I wonder whether he knew about him. Willis's book, written in 1664, was still used as a textbook when he was in Edinburgh as a medical student. So it's possible he saw it. Willis was the court physician at the end of the Civil War. He was in Oxford with that group of extraordinary people that essentially designed the Royal Society before it happened. He was a brilliant clinician, did his complete medical training in 2 years in the thick of the Civil War when Oxford was under siege. But he was also a great observer. You can see from the fact that his desk's littered with skulls. He dissected human brains, he dissected animal brains, and his writing is almost Darwinian. I mean, he very directly and explicitly compares the brains of different species, relating the differences to difference in behavior. So when he looks at the human brain, and this is Christopher Wren's drawing for his book, they worked together, Wren drew the pictures. This is the diagram of a human brain, indubitably, showing the enormous cerebral hemispheres here, and the cranial nerves, the optic nerves here, and so the base of the brain. And he said, okay, the cerebral hemispheres are enormously bigger than in another species. They must be responsible, therefore, for the things that human beings are particularly good at. That means memory, the superior faculties of the imagination, estimation, thought, and all of these things. Well, instantly by doing that, he made two major steps. One was this argument from structure to function. But the second was the assumption that the tissue of the brain, the stuff of the brain, did important things. For 1,500 years, people had thought, at least in Western science, that the function of the brain was entirely determined by the fluids inside it. And the tissue was sort of superfluous, except that it made the spaces for the fluid to live in. And here was Wren, by observation, concluding that can't be the case because the structure varies from animal to animal, so it must be doing something. Just a couple of crucial quotations if you'll allow me to finish. So the cerebrum, cerebral hemispheres, is the primary seat of the rational soul, the reasoning abilities. The animal spirits, I mean, he still thought that fluid did things, and animal spirit is the term that's applied to the fluids in the brain. Anima, from the Latin, meaning mind. The animal spirits, different spirits, for the various acts of imagination and memory, ought to be moved within certain and distinct, limited or bounded places. Localization of function. Very clear, the first statement. The different rational functions are localized in different parts of the cerebrum. And finally, this. These folds or rollings about, that's the convolutions of the brain, are far more and greater in a man than any other living creature. Yes, bigger and more folded. To wit, for the various and manifold actions of the superior faculties. You need a lot of brain to do all the things that we do. But they are garnished with an uncertain, as it were, fortuitous series. What he noticed was there were small variations from one person to another in the exact form of the folds of the brain. And his conclusion was that the exercises of the animal for the mind function might be free and changeable. There's a thread through our understanding of the brain from the 17th century, from this really seminal beginning, really, of a modern approach to neuroscience. There's a thread that runs through it from Willis to the phrenologists to Darwin and on to the modern day, which emphasizes that we cannot explain how our brains work without implying freedom and change. And it is that freedom of change at the level of the individual, which I think detaches cultural evolution from anatomical evolution. So a lot of what we are has not depended on a simple Darwinian process. It's depended on what Richard Dawkins would call memes, or cultural evolution, a kind of almost Lamarckian process. And of course, what's derived by learning and memory and cultural processes is not inherited in the sense of DNA. Of course it isn't. But it is inherited through language, through teaching, through education, through instruction, through libraries, in almost as solid a form as DNA. Well, but it's not, because it's more vulnerable. And my conclusion, the punchline, is that without explaining how our brains became big, it is certainly because we have them that we're able to do the things that we do. But it was not doing the things that we do now that led to the evolution of the big brain. There's a mystery still there. Well, if what we are capable of doing culturally depends on how we use the brain and how that information is culturally transmitted, then the good news is there's no reason to believe that it's over. There's no reason to believe that the lives of human beings in 500 years' time will be any less different from our lives than our lives are from those of people living 500 years ago, before science, technology, and all of those good things. The bad news is, because the mechanisms of transmission of this aspect of human ability do not have the organic rigidity of DNA to make sure they happen, they depend on what we tell to each other, the books we keep, the Googles and the internet and all these resources of information, and universities and schools. They are incredibly vulnerable. A single generation without access to the information of others and the accumulated wisdom of others would quite literally be back in the Stone Age. So we have clawed our way up by this amazing transmission of cultural stuff. It promises much more in the future than it's delivered so far. But we should protect it and guard it carefully, because it is much more vulnerable than our DNA.